I'm Stephen, and today I'm excited to be talking to you about the sponge model of cellular signaling. Now, much of cellular signaling in the body revolves around a family of proteins that we call the G-protein coupled receptors, or GPCRs. Now, GPCRs are extremely important in a variety of physiological pathways in the body, ranging from metabolism and neurotransmission to the five senses and cell growth, which relates to cancer. So it's not surprising that G-protein coupled receptors are extremely important pharmacologically. In fact, they comprise the targets of about a third of all pharmaceuticals on the market right now. So really, it's quite critical that we understand how they work. And so it's not surprising that they form a multi-billion-dollar industry in terms of studying them and understanding how they work. In fact, you probably recognize a lot of these drugs here on the left, which are all designed to target G-protein coupled receptors. Now, specifically, how do GPCRs work? Well, GPCRs are transmembrane proteins, meaning they span a cellular membrane here, and you can see in both of these images the GPCR is in blue. So there are 7 transmembrane helices all clustered together, and in general they will work as a ligand, or a small molecule will bind to the GPCR, triggering a conformational change, which opens up a binding cleft for a protein called a G-protein to bind. And once binding, the G-protein becomes activated so that it can go and perform other tasks in the cell, and hence why it's called a G-protein coupled receptor. Now, in general, one GPCR is capable of activating a bunch of G-proteins up until the point that another protein called arrestin binds to the GPCR and stops the signaling cascade. And so the arrestin is the termination point in the GPCR signaling. Now, this GPCR shown here on the right, this GPCR is called rhodopsin, and it is the visual protein that you're using to see with right now. And so it's responding to photons and allowing you to perceive that in your brain. And this protein, rhodopsin, is actually the GPCR that I'm studying as a model for the family of GPCRs. My specific project is that I'm looking at the role of water in the activation of G-protein coupled receptors. And you might ask, why water? Our bodies are 70% water, we've got water in all of our tissues. Why would water play an important role? Well, we think that water plays an important structural role in the activation of GPCRs. And the reason that we think that is, well, a few years ago, my lab performed a computer simulation of rhodopsin that showed that within 2 microseconds of the rhodopsin's initial excitation by a photon of light, there was this huge flood of water into the interior of the protein. In fact, there were over 70 water molecules that were flooding into the rhodopsin core. And this was really astounding to us at the time. We were wondering whether we could experimentally verify it. And if so, is it important? If we withhold water from going into the rhodopsin, does that prevent it from functioning? So that was what was motivating this project. And so we endeavored to experimentally study that. And the way that I did this was using the technique of osmotic stress. So if you're familiar with osmosis, it's the movement of water down a solute concentration gradient. So basically what we're doing here is we're introducing these polymer osmolytes, polyethylene glycol, or PEG, which behaves as a thirsty osmolyte that sucks water out of the rhodopsin. And by doing that, we can dehydrate rhodopsin and then measure how active it is using spectroscopy. And so by doing that, we can actually see how osmotic stress and osmotic dehydration is affecting the activity of the rhodopsin. And we actually use different sizes of the polymers, both large polymers and small polymers, anticipating that the large polymers will behave more like ideal osmolytes that dehydrate the rhodopsin, while the small polymers can actually penetrate inside, bringing water with them and having a hydrating effect on the rhodopsin. So we were interested to see how these different sizes of osmolytes might affect the activation of the rhodopsin. And lo and behold, our results confirmed our hypothesis. As we dehydrated the rhodopsin with these very large osmolytes, it had a very inactivating effect on rhodopsin. So here on this graph here, the y-axis gives the fraction of active state rhodopsin, and the x-axis gives the pH. So it's active rhodopsin as a function of pH. And what we did was we measured how this pH titration curve shifts under the presence of these different osmolytes. And what we saw was that by dehydrating the rhodopsin with large osmolytes, you systematically get lower fractions of active rhodopsin. So if you look right here at physiological pH, dehydrating the rhodopsin with large osmolytes drastically reduces how much is activated. But on the other hand, if we add small osmolytes that can penetrate inside, they're going to actually have a hydrating effect, and that shifts the equilibrium forward to more active rhodopsin. So we get very drastic differences depending on whether we're hydrating or dehydrating the rhodopsin with small or large osmolytes. And so this was really exciting because this effect was so huge. In fact, it's on par with some effects that you see with pharmaceuticals designed to modulate GPCR activity. But here we're doing it with water alone, not drugs, but just water, plain old ordinary water. So that's really exciting, and it's so exciting, in fact, that we decided to come up with a new model to explain this. And this is a model that we like to call the sponge model. Now in the sponge model, rhodopsin behaves like a sponge. In the inactive state, it's like a dry sponge. And in the active state, it's like a swollen, hydrated sponge. So just by varying how hydrated the sponge is, or how hydrated the rhodopsin is, you can vary how active it is. And as you can imagine, this has a lot of pharmacological potential, in that we imagine that we could vary the activities of GPCRs just by osmotically dehydrating them, or changing how prevalent the water is nearby them. So we're very excited to see where this could lead in terms of pharmacology. And with that, I'd like to thank the Brown Lab, which has been my research home. Thank you.